Next, we are going to hear from Professor Chris Contag, who's going to give us an update on IQ. Chris is, of course, the director of the Institute of Quantitative Health Sciences and Engineering. Chris?
So when Satish asked me to give you an IQ update, I, of course, had to say yes, but I suddenly realized that I didn't have any data. So I had to do an informal survey, and I'm happy to report that everybody in this room has an above-average IQ. We also use the name IQ for the acronym to describe the Institute for Quantitative Health Sciences and Engineering. The reason we use this is because if you just use the first letters of each word, it would be iQueasy, and we didn't want a medical engineering building named iQueasy, so we call it IQ. We have a simple mission to develop the tools, technologies, and knowledge to improve the health of people such that we can ensure 100 years of health for 100% of our people. That's the simple goal. I'm the director of the Institute, but also I chair the Department of Biomedical Engineering. And if you look inside this building, it has a beautiful architecture. 2 years ago, when we started this project, I was sitting down in one of these chairs you see at the bottom of the picture, and two people were walking through the building. And one of them said to the other, "What is this building?" And I was kind of interested in what the response would be. I thought, someone's going to say, "This is an amazing place where incredible science happens." This was their answer. Just a big glass, empty building. So I knew my mission. My mission 2 years ago was to fill up this building with incredible science and give it a name. So now the building has a name. It finally has a label on top, IQ, so at least those people walking through the building will say, it's this big glass, empty IQ place. So when in programming this building, we had to come up with themes that would tie the building to the rest of the university. And we kind of had a choice at the very beginning. We could have had one theme that would touch many parts of campus, or we could build on several themes that would tie into many programs across campus. My vision was to create a hub with spokes that would touch every project and every program that's going on campus. These are the 8 themes we came up with. The building has 4 floors. Each floor has 2 large laboratories. And so it made sense to have 8 thematic areas. These are the themes that are listed here in the order from the first floor to the 4th floor. And they're themes like systems biology, which builds on the huge investment from the VP's office in computational genomics. Bioimaging builds on the program in radiology and programs in physiology and pharm tox. And all of these themes are selected intentionally to build on existing programs. But we also had to house the Department of Biomedical Engineering. We could have done that in a couple of different ways. We could have put it on 1 floor or 2 floors and had an isolated group of investigators. But more importantly, or more effectively, we could have put engineers on every floor, in every lab, in every part of the building. That way we have an integrated set of scientists working in areas that are caps on top of existing programs. So chemical biology that builds on chemistry, chemical engineering, and pharm tox has engineers applying those tools. And that was the vision. So in those 2 years, we've gone from 0 to over 200 people. We have 240 faculty, staff, students, including 30 new BME graduate students over the last 3 years. And we just launched a recruitment for the next 12 graduate students. We have a total of 60 graduate students representing many programs on campus, from chemistry to BMS and others. There are currently 37 faculty in the building. We have a target of 50 faculty at the time of completion, and we represent 17 different departments and 6 different colleges. This group of 37 faculty have published 100 papers in the last year and brought in $20 million in federal grant support since the inception of the building. This image is a picture of one of our seminars that we call Brews and Views, and Steve Hsu is featured here with Len Fleck discussing the topic of whether we should genetically engineer our grandchildren or not. To support the research activities in this building, we built research cores on each floor. As I mentioned, bioimaging is a key part of it. Tools like this make our animal models transparent to scientific investigation so we can watch metastasis in real time, we can track disease, and better improve our therapies and our interventions. We teamed up, of course, with EFRIB, as was mentioned a moment ago, and I'll go into some detail in the next slide. In radiochemistry and nuclear medicine, this is a nice bridge between EFRIB and radiology. We count cells with tremendous sensitivity, another partnership with PharmTox. We developed a microscopy core. These are the glands in a mouse uterus at the time of embryo implantation. Amazing images being created by faculty in the building. Biostatistics looking at 300,000 parameters in a given individual over a 3-year period. A program in stem cells so that we can better regenerate and renew tissues. Genome editing so that we can edit those cells to do what we want them to do when we want them to do it. And a program in nanotechnology and one in precision nanomedicine, which would be driven by Anna Moore and her team as they move into the next building, which is going up next to ours. When we set up the facility, we had a basic principle. Culture is important. We need to be collaborative, integrative, we need to be forward-thinking, and I love this quote from Andy Grove, who said, "Culture eats strategy for lunch every day." We can have the best 5-year plan, but if we don't have the right culture, we won't achieve our goals. The IQ building sits in a group of buildings that we're calling the Biomedical Research Complex. This is a venture of many different colleges on campus really pushing the envelope of biomedical research. And as I mentioned, the new building for integrated science and technology for precision health is the one that will be added to this group. We work closely with nursing and the people in life sciences, and of course radiology and clinical sciences with the renovation of Clinical C as the animal facility attached to our building. The intent at some point in time would be that all these buildings are connected by bridges. Right now there's still some places, some gaps, such that we can move people and projects throughout this biomedical research complex in a thoughtful way. The whole program is based on building bridges. We heard about EFRIB, and we're building bridges to EFRIB with some of the isotopes they're developing, and that's a partnership, of course, with radiology, but Kurt Zinn was recruited to the IQ Institute to really build a radiochemistry program that ties us in through radiology and takes novel tracers into the clinics where people can use them to really understand disease and guide therapeutic intervention. This is just one example of the bridges we intend to build. IQ is supported by 3 colleges, engineering, natural sciences, and human medicine, and they tie us to multiple departments in engineering, multiple programs in business, in animal science. The College of Natural Sciences and Human Medicine ties us into microbiology, chemistry, PharmTox, neuroscience, and biochemistry and molecular biology. And we have a number of departments in medicine represented in the building, and we have a number of programs that we tie to, including into plant sciences, and I mentioned EFRIB, osteopathic medicine, veterinary medicine, and ag bioresearch. We're currently putting together a grant to try and fund a facility for large animal research with state-of-the-art PET-MR systems to really drive nuclear medicine and imaging. We've discussed in detail with many of our healthcare partners how we can improve their healthcare, their health delivery, how we can work with them to test our new therapies, and having McLaren on campus will make a tremendous impact. This is an artist's rendition of what that hospital would look like. The hospital, when it goes on campus, will take a net reduction in beds, which means they have the philosophy that healthcare will be delivered at home. That means we need better wearables, better sensors, better monitors, so that patients can stay in their home and not go to the hospital. We'd like to work with all of these healthcare systems to do that, including with Mary Freebed and their rehab programs, and integrate our genomic medicine into all of these programs. We're addressing a lot of very edgy issues. And so when I first got here, we started talking to the folks in the Center for Ethics in the Humanities and Life Sciences about how we can address these issues in a thoughtful way. So we put together a seminar series that we call Brews and Views, where we have one humanist and one scientist discuss really important issues that we're addressing in the building. The first one was called the Porcus sapien, or the humanization of livestock. Should we grow human hearts in animals? Should we use those for regenerative therapies? And each of these topics pushes the envelope, but addresses some key issues that we need to deal with as we advance biomedical research. We set this up as a discussion between a humanist and a scientist, and we asked them to publish a paper in the online blog called The Conversation, and this is one example of a publication that grew out of the discussion on whose fault is it if your implant fails and changes your behavior? Is it the manufacturer, the person who installed it, or is your change of behavior your responsibility? A really important issue. We've also launched the 10 Million Genome Project, which is the idea that genomic medicine should be a part of our normal health care. Our electronic medical records should include our genomic data because we can make predictions on risk. And I read Steve Shu's paper about a year ago talking about predicting human height from the genotype, realizing that this was the first step toward making really important predictions about human health based on our genome. We should be able to predict risk. We should be able to predict the risk of getting cancer when someone's 10 years old from their genome or when they're born, and we should be able to use that data to guide health care and make genotyping the standard of care in the state of Michigan. 10 million comes from the fact that Rick Snyder in the State of the State a couple of years ago mentioned that he wanted to increase the population of Michigan back up to 10 million, where it was 10 years ago. And so we thought 10 million human genomes, we should all be genotyped. So that was the root of the name. This all fits into several revolutions in medicine. We've heard about personalized medicine, which is this idea of individualized monitoring and individualized care. That's kind of morphed because of the cost of individualized care into precision medicine, the idea that we should be targeting the molecular basis of the disease or the molecular etiology. We'd like at Michigan State to take this the next step, to take this to precision health. If we can predict risk, health risk, in the early ages, we should be able to intervene early and maintain health rather than treat disease. That's the notion of precision health, and Anna Moore was recruited here to run a program in precision health and really extend that across all medical schools, nursing, and veterinary medicine to really change the paradigm from treating illness to preserving health. The reason this is so important is represented beautifully by this graph that was published by a colleague of mine in Wired magazine. He thought it was important that the general population know about the importance of early diagnosis. If you look at the far, at stage 1, this represents the early diagnosis and prognosis of cancer. If you look at stage 1, diseases detected in the stage 1 have an 80% 5-year survival rate, regardless of whether you're looking at pancreatic, lung, ovarian, or prostate cancer. If you wait till the disease is stage 2, 3, or 4, the 5-year survival rate drops below 10% for some, below 20% for most. Early detection is key. Prevention is important. We need to intervene early with our diseases, so we need sensors, monitors, and tools that allow us to detect disease early so that we can intervene early. This has led to a number of companies growing out of the faculty laboratories in IQ. These are some examples for new startup companies growing out of the Institute. And we feel that the only way that our tools and technologies are going to get into the hands of the people that need them is to make them available through commercialization. So we've been working with the MSU Foundation. I think the MSU Foundation funds 4 of these companies in part, and all of them are in very early stages. But we think it's really important that we take our tools into the clinic and we take them into the marketplace so that they can be used by the people who need them. Why do we do this every day? Why do we jump out of bed and say, I get to run into the lab and work today? Because everybody deserves a century of health. Everybody deserves the opportunity to have a healthy life for 100 years. And that's the mission of our institute. Thank you.